MF. All right, so today I'm going to talk about an alternative way of analyzing your single-cell transcriptomics data. And it's basically within the framework of exploratory data analysis. So this is the outline of the talk. In the beginning, we'll just discuss very briefly what the different sources of variation and let me get rid of that window. Different sources of variation in single-cell RNA-seq data are, and what are the different ways that we can think about categorizing them. Then we'll talk about different computational approaches for studying these sources of variation. And then I'll give you a case study of healthy rat liver atlas and how we use basically this exploratory data analysis approaches to understand novel biology about this new map. And then I'll briefly discuss matrix factorization methods, and I'll talk about factor rotations. And then at the end, I'll mainly talk about how to interpret the resulting factors, and we'll end the presentation with some tips and takeaways. Let me find my cursor. All right. Okay, so there are different ways to think about sources of variation in single-cell transcriptomics maps, and I've tried to show you guys some different categorization in this mind map. So one approach I find useful for thinking about modeling variation in single-cell transcriptomics data is to categorize them based on whether or not there's sample level attributes, whether or not there's cell level or cellular level factors or attributes, or whether they are gene level factors. And each of these categories can have some biological examples and technical examples. And what do I mean by that? For example, the sample level covariates or sources of information that you could be interested in study could be the age, sex of the patient, strain, disease, ethnicity, experimental condition, time points, and so on. The technical examples could be batch, the single-cell capture technology that you've been using, for example, whether or not it's plate-based or droplet-based, or for example, different dissociation protocols that you use for preparing your samples. Some examples of the cellular level factors could be, for the biological ones, could be cell type identity, cellular states, cell cycle stage, or like pathway activity of individual cells in your sample. And some of the technical examples are library size, number of expressed gene, mitochondrial gene fraction, and contamination. So basically, what we want to understand is that how much each of these different covariates are explaining the variation in our data. And when we find the major sources of variation, how can we map that information into specific cell types and specific genes so that we would be able to learn about the biology of our data. Final category in this mind map here is the gene level factors. So what I mean by that is that sometimes you might have some information about your genes rather than the cells that you're studying. And that could be pathway information or different gene set information, for example, transcription factor gene sets. And we're all familiar with gene ontology, Reactome, MCDB, and CHIA is a transcription factor gene set. And I'll just briefly explain that another way to look at this is that you can think whether or not any of these examples that we talked about are unknown or known. So sometimes, this information could be provided as a metadata, or you could directly calculate it based on your data. For example, library size, you can easily calculate that based on the given data. But sometimes some of these information are just unknown. So for example, you might not know the cell type identity of the set of the cells that are included in the sample. And another way to look at this is whether or not these covariates are discrete or continuous. So discrete would be, for example, male or female. And for continuous, you could have like gradients of cellular states. And one of the reasons that I wanted to emphasize this is that when you're thinking about mathematical modeling or doing any sort of computational modeling on your data, it's important to know which of these categories you're trying to focus on. For example, what I'm showing here in this matrix here is that we have cells as rows and genes as columns, and any of the cellular level or sample level information can be encoded in a matrix like this as a metadata. But if you're trying to incorporate gene level information in your analysis, it would look like something like this. So even the dimensions of these two matrices would be different. So before starting your analysis, it's important to have a well-defined question based on the metadata and information that you have. Okay, so what are the different methods for studying each of these covariates? So I think one useful way to think about this is to kind of split this into supervised and unsupervised approaches. And what I mean basically by supervised approaches is the differential expression analysis, which we comprehensively discussed last time. So Brandon walked us through the different differential expression approaches that are out there. What are the current computational challenges with each of them? But basically, these methods are useful when you have a specific direct query in mind that you want to look for. For example, you want to know what's the difference between these two specific cell types in my sample. Or imagine you have multiple different samples and you would want to know what's the difference between the T cells that are in condition A and the T cells that are in condition B. So you have a very well-defined question to ask. So as, again, Brandon explained last time, currently we have two general like statistical models that are kind of very well fit for answering these questions. And the first one is generalized linear mixed models. So they've been developed to account for the pseudo-replication bias of single cell data. And what we usually do is that we model sample as a random effect. And some of the popular implementations are Nebula and MAST. So if you're using droplet-based data, usually Nebula is preferred. And another approach is to use pseudobulks. And then you would be able to use traditional bulk RNA-seq methods for performing this differential expression analysis, and they're usually based on generalized linear models. So some examples are DSEQ2, Womb, Lemma, and many others. So the second approach that is the focus of the talk today is the unsupervised way, or you can think of it again as the framework of exploratory data analysis. So what we're going to do in this framework is to apply some sort of matrix factorization method to the gene expression data, and then come up with different ways to interpret the learned factors given the metadata that we have. So I will explain what I mean by matrix factorization in a little bit. So don't worry about that. But just before going into the more details, I wanted to explain why would you be interested in this second approach, the unsupervised approach? Why bother solving the problem in an unsupervised way when you already have some metadata or annotations? So there are different reasons that you would like to do that. First of all, as again we discussed last time, the current implementations of differential expression suffer from many different computational challenges. Almost all of them are suffering from type 1 error. So we discussed pseudo-replication bias, double dipping, and distribution misspecifications last time. So the differential expression problem has not been yet figured out for single cell data. So the results that you get might not be very accurate. The second reason is that as the metadata increases, so when you're handling very large data sets with a lot of different metadata, the number of combination of queries can become overwhelming. So you might get overwhelmed that what should I be even looking for? Because there's just so much information, there's so much interesting questions that you could be looking into. So an unsupervised approach can be a useful tool to quickly identify the strong signals in your data without needing to specify each of the queries individually. The third reason is that some of the covariates might be unknown. So you might just not have some of the information as a metadata. For example, I don't know, some of the patient information might not have been provided for different privacy reasons. Or sometimes some of the covariates might not be well defined. So for example, cellular states, we don't really understand them yet, or different gradients that might be interesting to study. And the fifth reason, which is kind of the main focus of the case study that I'm going to walk through, is contamination. So contaminations are usually, at least at this stage of the field, they're kind of difficult to model directly, and they can potentially mask the biology and lead to inaccurate results when you're approaching the problem using the direct queries. Okay, let's go.
I have a question, if I can. We can talk in the middle, right?
Yeah.
Okay. Out of everything that you presented here, is this all linear combinations of these features? And do you think it's worthwhile to look at non-linear aspects of the relationships between these covariates?
Yeah, that's a good question. The focus of this talk is only on linear approaches, and the main reason is interpretability. So depending on the data and the patterns that might be in that specific data, nonlinear methods could be useful, yeah. But again, majority of these nonlinear approaches, for example, for the factor decomposition approaches, we have a lot of autoencoders that are out there, but they suffer from lack of interpretability. So it's very hard to map the information that you get into specific genes that represent that variation. So that's the major reason that, for example, in this case study that I'm going to go through, or in many other projects, we chose linear methods over nonlinear, but it can be worthwhile to just apply one of the nonlinear approaches to the data and see if some interesting pattern comes up. But again, it would be hard to interpret. I'm going to recommend a few nonlinear approaches at the end of the talk, so we can also discuss this a little bit later towards the end.
Okay.
Perfect. Any other questions? Sure. I don't think so. Okay. So the case study that I'm going to talk about is the Healthy Rat Liver Atlas. So in collaboration with McFarland Lab at UHN, we were trying to generate the first Healthy Rat Liver Atlas. And the reason that we were interested in this map was that rat is used as a transplantation model. And two of the main strains that are used for modeling transplantation processes and rejection and tolerance are Lewis and Dark-Agouti strains. So basically, they wanted to know what's the difference between Lewis and Dark-Agouti, because throughout the transplantation, these rats tend to behave differently. So that was the biological question that we wanted to answer. So here in this chart, I'm basically showing you the traditional approach for constructing a map and like doing very basic analysis on it. So you start with quality control and normalization. You cluster your cells and visualize them, and then you identify markers, perform pathway analysis to gain some functional insight. And at the end, you try to annotate your clusters, and then you end up with your map. So as we were going through this process, we came across multiple different challenges. So in this example, some of the cell types in the liver, such as hepatocytes and cholangiocytes, are particularly sensitive to physical stress and known to get damaged throughout the dissociation process. And when they're damaged, their mRNA gets released and contaminates the other cells. So the contamination caused by these released mRNAs is called the ambient RNA contamination. And basically, as a result of this ambient RNA contamination, the marker list that we get didn't really make that much sense. So we couldn't perform annotation other than the marker genes. No matter what pathway analysis you performed on this set of markers, none of the pathway analysis methods would work. And maybe even more importantly, the whole point of this project was to be able to compare different two strains. But because of this contamination, we couldn't really perform between strain comparison, because no matter what cell type we would look into, all the top markers would be hepatocyte related genes. So we had to kind of figure out a way to parse out the biological and technical signals within the data. So before talking about the method that we use on here on the right, this plot showing the albumin expression over the map. So albumin is one of the main hepatocyte markers, and you can see that no matter what cell type you're looking at, you can see a very high expression of albumin, which is caused by the ambient RNA contamination. Okay, so all these challenges highlighted the need for a systematic approach to dissect the biological and technical variations of the data from each other. So matrix factorization is a class of unsupervised techniques that try to reveal the low dimensional structure of the data while preserving as much information as possible. And how it does that is that you basically start with your matrix of interest, and then it kind of tries to split that input matrix into two smaller and simpler rectangular matrices, so that you can have you can basically study and interpret each of these matrices, which are simpler, individually and in an easier way. So you start with your data, it tries to split the data into simpler entities, and then you study those simpler entities and try to make sense of them. So there are a lot of different matrix factorization methods out there. I'm going to go through them now, go through the famous ones in a couple of slides, but basically the general idea is the same for all of them. So what you end up with is one matrix that I'm going to call the loading matrix. So the loading matrix represents the relationship between genes and factors, and the score matrix represents the relationship between factors and samples. So this loading matrix can be used for gene set discovery, pathway analysis, and so on. And the score matrix can be used for clustering visualization. Okay, so how are these two matrices actually learned? So again, as I mentioned, there are a lot of different matrix factorization out there, and the main difference between them is that they have different assumptions about the input data and also different assumptions about what informative means. And the way that they mathematically implements that is that in order to be able to split the data into these two smaller matrices, like many other machine learning algorithms, they convert the problem into an optimization problem. So they want to have a matrix. They want to find these two, basically approximate these two loading and score matrices in a way that their multiplication would be very similar to the input matrix. So in order to implement that mathematically, they kind of try to minimize the distance between the multiplication of these two matrices and the original matrix. And people have very different like distance matrix that they can use. Some people use squared, some of the squared error as a distance. You could use Frobenius distance as a measure, and there are many different ways to mathematically model that. And each of these could lead to like different matrix factorization methods, but that's the general idea. Okay, so what are some of the common matrix factorization methods out there? So I would say by far the most popular one is principal components analysis, which probably all of you have at least seen once in your life. So what basically it tries to do is it tries to identify this low dimensional representation of the data based on some new accesses that are able to maximize the variance of the original data. So usually the columns of these new principal components need to be not usually, basically that's the mathematical requirement that they need to be orthogonal to each other. But one problem with PCA is that maximizing this variability captured in each of the factors can sometimes mix the signals from multiple biological processes in a single component. And that can potentially make the interpretation of the resulting factors difficult. And I'm going to talk a lot about this in the next few slides and the ways that you can overcome that. So one of the other super popular matrix factorization methods, specifically in genomics, is non-negative matrix factorization. So what NMF does is that it constrains all the elements of the two, basically both the input matrix and the loading and the score matrices to be greater than or equal to 0. And this non-negativity constraints makes the representation of the data purely additive. So there's basically no way to remove the signal throughout learning this set of new low dimensional spaces. So that kind of usually leads to sparse representation of the data that's easy to interpret. Again, that's like interpretability that is kind of super important for us when we're studying biology. And so again, it's usually easier to interpret, and that's like the non-negativity constraint, which is usually true for biological data. And also this sparse and interpretable learned factors has made it super popular in the field of genomics. And what I'm showing you here in these pictures this is from the original NMF paper. So they have applied NMF and PCA to an image data. And what you see here, these two are the loading and basically score matrices. And as you can see, it's making sense of most of the factors in the PCA, score matrix is very difficult. You can't really, it's kind of like, it usually tends to learn some global features. But for NMF, if you look at these patterns that have been identified, you'll notice that it's kind of identifying different, very individual facial features. Like some of them could represent the eyes, some of them could represent the nose, some of them are eyebrows, or some of them are the lips. So it kind of decomposes the initial image into like these little parts and edges that are very easy to interpret. So that's kind of our goal here. And so the third popular matrix factorization method that I'm going to talk about is ICA, or independent components analysis. So the assumption of ICA is that it kind of tries to identify sources of identify independent sources of variation. So so the factors that are going to be identified are going to be statistically independent from each other. And again, it solved this the optimization problem that I was talking about, based on minimizing the total mutual information between the K estimated components. And here, I'm kind of showing you an example of ICA versus PCA on an image data. So what we see here is kind of a mix of 3 different images. And on the right, what we're seeing is that when ICA is applied, you know, in the 3 first components, sorry, let me get. You can see that the components are capturing meaningful images, but with PCA, it kind of seems like it's averaging some sort of different averages between these 3 images. So they're, again, very hard to interpret and figure out what each of these components individually mean. So there are a couple of tips and notes that you should have in mind when you're using these methods. So solutions from both ICA and NMF may vary depending upon the initialization of the algorithms. So it's important to ensure robustness of your factors before starting the interpretation. So this is not true for PCA. PCA has an exact solution, an exact unique solution, and is solved analytically, while these methods are solved using stochastic gradient-based numerical approaches. So that's basically why you don't have to worry about this with PCA. And unlike PCA, the learned components in both ICA and NMF are assumed to have equal weights. So if you've used PCA before, you know that you usually use elbow plot and scree plot to like choose the number of principal components that you're looking into, and they're usually sorted, like the first principal component has the most information. This is not true for ICA and NMF, so they have equal weights. And the last point about NMF is that there are some Bayesian techniques and Bayesian implementations of NMF, and it's been proposed in the literature that these methods might have more robust loading matrices compared to gradient-based ones. So maybe consider using Bayesian-based NMF approaches. And I'm going to briefly mention a single cell specific one at the end. So another way, so again, we're talking a lot about interpretation, right? So we care a lot about interpretability of the learned factors, and I will discuss actual pipelines for interpreting the factors from the next slide. But before moving into that, I wanted to discuss an approach that you can potentially improve the interpretability of the learned factors, and especially for PCA. And these methods are rotations. So rotations are basically any sort of like approach that you would like apply some rotation to your loading matrix in the space and rotating it in a way that the resulting space would be more interpretable and easier to study. And I'm going to show you an example here. So I would say that the most common rotation that is out there is Varimax, and it's a relatively old method. And again, it brings the loading matrix closer to a simple structure, and it's mainly applied to PCA. So it's kind of not super common to be applied to other matrix factorization, but it could be. And basically the way that it achieves this more interpretability is that for any given factor, usually you end up with the factors that have lower, like there are some of the features on that factor, like a few of them have very high loadings and the rest of them are basically loaded close to zero. So you can easily associate each factor with specific number of genes. So instead of having like a lot of genes with like some middle score, you end up with, you know, some factors that have high scores for a few genes and the rest of the scores are close to zero. And I'm kind of trying to show that in this, you know, simple example right here. So each dot here represents a gene or a feature. So we have factor 1, you can think of it as principal components number 1, and this is factor number 2. And these are basically this is the loading vector or the loading matrix that you would look into. And you can see that like it's very hard to say that like factor 1 is only associated with like gene 1 or gene 2. It's kind of like have some middle score for most of them. And the same is true for factor 2. And when you rotate these axes, oops, let me go back. You end up with a loading matrix like this. So like you can see that for the first 2 genes, the score is near 0, and the last 2 ones are high, and similar pattern for factor number 2. So you can associate factor number 1 with gene 3 and gene 4, and you can associate factor number 2 with gene 1 and gene 2. So this is basically what I mean by simpler structure. And this is just another kind of cute example that Tallulah suggested. So if you're trying to like learn features of a horse using PCA, what PCA does is that it tries to maximize the variance, right? Then basically finds the factors that are able to maximize the variance of the original data. And these gray axes would be PCA learned factors. And if you apply Varimax to PCA, you will end up with this red axes, which are kind of more specific rather than trying to maximize the variance. And you can like, if you split the horse using these 2 cuts, you can have a much easier time like figuring out what each part represents, rather than splitting the horse using this gray cuts, basically. Okay, so what does it look on actual single cell data? So when I'm talking about being hard to interpret, I mean some pattern like this. So each dot represents a cell here, and they're colored based on their cell type or cluster. So you can see that's very hard to figure out what principal component number 1 and 2 are representing before applying the rotation. And this is what you get after applying the rotation. You can see that like the signal is a lot more clear, and you can easily associate Varimax rotated principal component 1 with hepatocyte population, and Varimax component number 2 with the macrophage population. So this is like an ideal interpretable factor that you would like to get. Okay, so
I have another question, if I can.
Sure, go ahead.
It's Gregory Schwartz again. My second question. So a lot of times if we do PCA and we can get something like this, and sometimes it may be from batch, we don't know, that we would get this like spread or these volcano kind of PCA, you know, shooting out or not, but we can see stuff like this, right? And so we have here these 2 different axes, and you're visualizing now the macrophages and hepatocytes. So now if you want to see what the other cell types are, like, because they're all just clumped together, do you just like, you do this manually? So then you say, okay, what's the 3rd and 4th, and then 5th and 6th, and plot each one, because you can only do 3 at a time, right? At max, right?
Yeah, that's a great question. That's basically the next slide.
Oh, never mind. Sorry. Okay.
Yeah, no worries, no worries. It tells me that the flow of the slides are good. So these questions are already in your mind. So yeah, one way is to do that manually. And here, for example, you could visualize, instead of like Varimax number 1 and 2, you can visualize Varimax number 1 and 3, and then go through each of the components individually. But there are, like smarter ways to do that, and I will briefly go over the different approaches that you can take in the next few slides. Okay, so let's talk about interpretation. So before going into the details, I just want to mention that performing interpretation can be done using both the score matrix and the loading matrix. So you can either start with the score matrix and then figure out the genes using the loading matrix, as I will explain in a little bit, or you can just start with the loading matrix if you don't really care too much about figuring out the specific cell populations or something. So if you want to directly move to the genes, you can also do that. But if you're going to start with the score matrix, this is the way that you would do it. So what you want to do is to match the factor scores to a covariate of interest. Again, a covariate could be anything, could be like age, could be strain in the case study example that I was talking about, could be cell types. So in this example, we were interested in identifying the cell identity program of hepatocytes or macrophages. So it can be anything, any sort of like metadata, or it could be even batch. So you might be interested in understanding some information about where the batch effects are coming from, and which genes are mostly affected by these batch or any sort of other technical variation in the data. So, again, you have some covariates of interest in mind, and you try to match the factor of interest with this covariate. So you can do that using visualization, as I was showing you in the last slides, but it's kind of manual and it's hard to do. Like, it takes time. Like what you would have to do is to kind of similar to this example. Another example that I'm showing here is that you would individually visualize your factors. For example, I'm showing Varimax number 10 here and Varimax number 1, and this is the rat data, by the way. And I'm kind of coloring each of these dots or cells based on which cluster that they're coming from, and each cluster represents a cell type. So you can see that, or you can, represent the same information using box plots. So we have clusters on the x-axis and the Varimax 10 scores. And again, these scores are coming from this matrix, the score matrix that relates samples and factors. And you can see that, this cluster number 7, this pink population, is associated with this Varimax 10. You can also kind of project that into the UMAP and see where they're which part of the UMAP has higher scores, and you can see that it's this part, which based on our annotations, I know that this is a natural killer and T cell population. So basically this Varimax number 10 has captured the cell type identity program of T cells or natural killers. And so that's one way to go, to kind of visualize them individually. Another way is to kind of try to automate that process using other correlation approaches, which is like the simple way that you can do it, or you can train some machine learning classifiers on your data, on your factors. Basically what that means is that you use your factors as inputs to your classifier, and you would it's necessary here to use a machine learning algorithm that is interpretable. So you can look at the feature importance scores. And so you would try to basically predict the covariate of interest, for example, strain number 1, strain number 2, based on, so those covariates would be your labels, and you would like to predict those labels based on the given score matrix of the factors that you're trying to interpret. And then if you look at the feature importance scores, they would basically give you the match, like which factor is the best match for the covariate that you're trying to study. And I'm going to show you the feature importance scores of strain in the next slide, in the case of study example that I was talking about. Okay, so after you've been able to match a factor of interest to the covariate, then what you want to do is that you want to evaluate the genes related to that covariate using the corresponding loading vector. So again, this is the score matrix. We have samples, we have factors. Like here, imagine that this is like variance factor number 10. Then I would go into my loading matrix, look at where variance number 10 is. So here it's actually like number 1, but doesn't matter. It's the same idea. And then like these are the genes and their scores for that specific factor. So what this list basically similar to is very similar to the list of differentially expressed markers for that specific cell type that I'm trying to study, if the covariate is cell type. For example, if I look at Varimax number 10 in my loading matrix, the scores, and I sort the genes and look at them, like the order of the genes would probably be super similar to the order of the markers of this T cell or NK cell population if I look at my differential expression matrix. Okay, so you go to your loading vector and rank and select the highly loaded genes. Again, you can those and kind of study those genes if you're interested. They can be kind of followed up in experimental validations, or you can kind of have a more holistic view and try to perform pathway analysis or gene set analysis on that sorted list of genes and kind of figure out what biological function is that factor representing. And so here I'm showing you an example of matching factors and cell types using the loading matrix directly. I said that that's also another way that you could do it. So this is based on correlation, super simple. So each of these rows here represent the average expression of that specific cell type or cluster. There are some hepatocyte populations, lymphocyte macrophages, and so on. And on the columns, I have the factors. So these are Varimax factors. And you can see that, for example, this Varimax factor number 1 is matched with macrophage populations like Marco positive and CD50 positive macrophages, which are non-inflammatory macrophages. And what Varimax number 2 represents is the cell type identity of some hepatocytes, and so on. So for the example of using, machine learning models to kind of automate this process, here what we have is that, again, in that case study, I was interested in understanding the differences between the two strains, the Darko-Goody and Lewis strains. And what we did was that we used a random forest model, and random forest models or any other model based on decision trees, I would say would be a good option here, because you can, they're highly interpretable. You can easily access their feature importance values. And so there, again, there's a lot of literature on this. There are a lot of different feature importance scores out there, but here we were looking into mean decreased Gini scores. And again, what I have done here is that I've used my factors to predict the strain label of individual cells. And then I've looked at which of these factors were more important or more informative for the model to make that prediction. And what it shows me is that Varimax number 5 and Varimax number 15 have been very informative. And if I look into these factors, here I have Varimax number 1, just for the sake of visualization, on the x-axis, and Varimax number 5 and Varimax number 15 on the y-axis. And I've colored the cells based on their strain, the strain that they're coming from. And for Varimax number 15, you can see that, we have these two clusters of cells from Darko-Goody and Lewis that seem to be separated based on Varimax number 15. So as I will show in the next slide, if you look at what cell type these two clusters are coming from, they're coming from non-inflammatory macrophages, which was very interesting for the biology that we were trying to biology of the rat livers that we were trying to kind of figure out. And on Varimax 15, you can see that you're apparently saying some sort of global effect. So it seems to be able to separate out all the cells of Yarkaguchi and Lewis from one another. Okay, so how?
All right. Okay, maybe this will be my last question because I keep.
No
talking.
worries. Ask as many as you want.
Okay, great. So here in example 2, you are using a random forest model, which is not very interpretable, right? So you have the decision tree is very interpretable, but when you have many decision trees that are randomly generated that are cut, it becomes much less interpretable. And there are these feature important scores, but those are also like not, I guess, optimal because like they're basically you can do like a perturb kind of processing where you can, you know, take out a feature, see how, like that's probably what this is doing, and that sees how
not exactly. Yeah, that's one way that, you know, these methods work for figuring out, and that's valid too. You're basically using different, like decision tree would work too. You can try that out. Yeah. I would say that the results of these different models, because I've tested a few of them, doesn't really change that much. Isn't super different which one you use. Like you could also use like a simple logistic regression, right? And look at the coefficients that are learned, and that works too.
But so the question is that, like, you know, in theory, like, you know, like there are more and more interpretable kind of trying to get the interpretability out of these these black box methods. But like, so right now, like the idea is you could use these black box methods on interpretable algorithms in order to identify which components are interpretable. Like, is that a valid statement?
Are you talking about deep learning methods?
Well, that or like a random forest, right?
Yeah, I mean, but it's different, right? Like the amount of interpretability of random forests is still pretty different from deep learning models. Like
yeah,
deep learning are a lot more challenging to interpret.
You can use that feature importance, perturb, whatever. There are a few algorithms to do that with deep learning. So
yeah,
like,
I mean, like
you could use the same idea.
Yeah, I mean, there are people have done that. Like there is like Deep Tailor, I think, or like a lot of different approaches that people have tried to figure out try to come up with creative ways to interpret deep learning. But like my current understanding is that the field is not there yet. So like, and another thing to consider is scalability. As far as I know, all of these interpretability methods that are kind of available for at least autoencoders and variational autoencoders are not very scalable. So scalability also matters. Like it might take a lot of time. Again, all of those are valid approaches, and people have done that. There are like specific papers for single cell data that people have been trying to do that. But I would say that the mapping and the interpretability is a lot easier using these approaches. And this is kind of like a way just to match the factor with the covariate, right? So you basically don't need to do anything to figure out what genes are representing that specific factor. That's given. And the interpretability of like some of those, you know, like again, autoencoders, you would do like, you would have to apply these, you know, additional approaches like DeepLeaf, Deep Tailor, and like these other methods, just to figure out what genes are representing this, you know, like factor, like score factor, like cell-related score factor of interest, just to do the mapping between the gene and the factor. But here it's like the loading matrix and the score matrix are just given like very quickly. But what here we're just trying to do is to kind of, instead of manually visualizing each of the factors, which that's also a super valid approach if someone wants to do that, you could like visualize each of the factors and then like color them based on the covariate that you're trying to study, like what I'm doing here, right? So this kind of random forest model kind of just made that process a bit faster. It's not about interpretability of matching the genes, like there are like different layers of interpretability, I think, that we're talking about. Does that make sense?
Yeah, it does. I think it is kind of valid to like use the uninterpretable black box methods as kind of like a p-value to identify the interpretables results, right? Like you could argue to do that. I guess.
To identify the
like here, you're identifying I don't want to take up too much of the time here, but like
no, it's okay.
The fifth dimension here is the most important one, right?
Yeah,
So,
basically my problem with these, like, and it's not a problem, just like the only reason that I wouldn't use like a variational autoencoder as my first choice is that this loading matrix is not given. So it only gives me this. It gives me like sample and factor. So I need to do like more work to approximate this gene and factor matrix with different ways. And people are trying, this is again, active field of research. Like people are trying to figure this out. But as far as I know, hasn't been solved yet. And, but again, like some people have been trying to do that. Like you can even like, it has even been optimized for single cell data if you want to try it on your data. So again, by all means, try them as well. Like they're awesome. And again, these linear methods, like by definition, have the limitation of identifying patterns that should be linearly separable. So if they're not linearly separable, like PCA can't find it. So like more complicated patterns might be missed. But again, there are like trade-offs all the time, right? So I hope that makes sense. But I'm going to introduce like a couple nonlinear ones in 2 slides for you if you want to look at them in more depth.
Thank you.
No worries.
Can I ask a question?
Sure.
This might be a kind of a silly question, to be honest.
No.
But just looking at the previous slide, the correlation, I was wondering, like, is there a way to kind of, I don't know, supervise this process so where you can have the covariates as specifically, let's just say cell types, macrophage, hepatocyte, whatever. And you say that these are covariates, and the Varimax factors have to figure out exactly which gene, like ranking is specific to which cell type.
Well, we're kind of like doing the second part that we're doing is supervised, right? So just identification of the factors is unsupervised, right. But matching the factors and
So I'm saying that is there a way to do it backwards? If you have the covariates, or you have the different cell types, for example, and then you're saying when you do the PCA and when you do the rotation of the Varimax, can it rotate in a way to fit exactly where each Varimax factor is specific to one cell type or one particular?
Do you mean like doing the 2 steps in 1 step?
Yeah, or kind of like the other way around.
It's not I don't know why you say it's the other way around.
This might be a silly question.
No, it's not. There are other methods that kind of are similar to PCA and are supervised, like LDA is another option, like linear discriminative analysis. So it's basically like very similar to PCA, but tries to identify like again, factors or like eigenvectors that that are able to separate like the points of the data points based on the features based on the labels that you provided for. So LDA is an option as well. It's not, I would say, like, again, like interpretability would be an issue, and like you would miss the unknown factors because you don't always have all the metadata provided, right? Or you might be like missing some, or it might be some new biology that you could discover and you would miss if you like define your problem in that way. For example, it could be a new cell type that you didn't know about, or a new subtype of that cell type that you didn't know about. So that's like the shortcoming of those methods. But yeah, there are definitely some like some matrix factorization methods out there that try to tackle the problem in a supervised way. Yeah, no worries. Okay, so here I just wanted to show you some of the interpretation results of that case study that I was talking about. So again, I talked about this Varimax number 15, and I told you that these two cell types, these two like population of cells that Varimax 15 was able to kind of separate from one another were non-inflammatory macrophages. And if you map those scores into the UMAP, you can see that very clearly that they're kind of like aggregating here. And this population, if you annotate this map, you'll see that it's a myeloid cell or macrophage cell population with Marco positive, which means non-inflammatory. And here what I'm doing is that I'm kind of like evaluating the expression of the top 20 genes of that Varimax number 15 to figure out like what are the genes associated with this Varimax number 15. So these are like interesting markers that could be studied experimentally. And I also talked about pathway analysis and gene set analysis. So this is transcription factor gene set analysis. So again, in order to do that, I went to the loading matrix, selected the Varimax number 15 loading values, sorted the genes based on those scores, and like applied GSDA or any other interpretation, any other pathway analysis method that you might be interested in, visualized using Cytoscape. And this is basically the gene set enrichment analysis based on CHIA data set, which is a transcription factor gene set. So you can identify what biological functions are represented based on that factor of interest. And by interpreting and putting together all of these results, we kind of hypothesized that the myeloid population kind of indicated a higher baseline inflammatory states in the Lewis rat compared to the dark agouti rat, which our collaborators and Sai in the McFarland lab were able to validate experimentally. Okay, so as I was telling you guys, so we started 5 minutes late, so I can have 5 more minutes, right? There are a lot of different like methods similar to like Varimax PCA that I was talking about, or like the other traditional ones are out there, and they've been kind of developed for single cell studies specifically. I've kind of wrote down a couple of more popular ones here. So SC-CoGAPS is an NMF-based approach, and they have used Bayesian implementations, as I was kind of mentioning before, because of this stability of and robustness of the loading vectors that you get. This is FSCL-VM. This is also another factor analysis approach specific for single cell, and they use latent variable models to perform the modeling. But the way that they model these attributes is a little bit different. So they let you incorporate gene sets rather than like metadata, like cell-related metadata. So they use like gene ontology gene sets as the incorporated information. So NIFA is another single cell specific matrix factorization method that kind of try to mix ICA and NMF. So this is also interesting to explore. CNMF is consensus non-negative matrix factorization. So as I was explaining, one shortcoming of NMF is that you don't end up with a unique solution. So in each time you run NMF, you might end up with different factors. So basically what it does is that it kind of tries to come up with more stable factors by running NMF like 1,000 times and then clustering the factors and representing those as a more robust solution. And for the autoencoder ones that I've been talking about, this is one of the popular like nonlinear methods out there, like LDVAE. It's a linear decoder variational autoencoder. So it's again, it's a variational encoder deep learning based method. And this is basically me applying these methods to a subset of the data that I had and visualizing the factors that kind of represented this NK cell population. And if you apply all of these methods and kind of like try to cluster the factors, so here, like this is the correlation matrix. So X and Y labels are the same, and each line represents one of these learned factors for one of these different methods. You can see that majority of the like prominent cell type identity factors have been captured by all of these methods. But when you have like smaller populations, which means that the signal is less strong, you can see that I think only one of the methods was able to only one of the factor analysis approaches was able to capture that. And scalability is also another thing to consider. Like FSCLVM has a very interesting method, but it's like insanely short. I did an scalability analysis on it, and it, like on large data sets, it could take like 50 days to finish running. And again, the interpretation might be a little bit difficult for some of them. And this is like an inherent problem, I would say, of this factor analysis approaches, because you need to, for many of them, you kind of need to figure out K, which is the number of factors that you want to learn. Okay, so I'm going to finish with just very quick tips and takeaways. The first one is that be careful about technical and batch related signals that you get, like something that is useful to do when you're trying to interpret your factors is to kind of try to correlate them with some of the like traditional QC covariates like library size, contamination estimation, cell cycle stage, like mitochondrial fraction and so on, and see if it's like highly correlated with those ones, which means that it's representing technical variation rather than biological. Another point is that multiple signals might be captured in a single factor. So that's something that you should always consider, that one factor, like ideally we would like to get like one-to-one mapping between covariates and factors, but that's not always the case. And so this is just an example of how one of these factors were capturing contamination and strain-related variation together. And one way that you can do computational validation of your signals of interest is to kind of use independent data sets to project the signal and basically the factor that you've learned into. So you could select the like highly loaded genes and kind of convert that to a gene set and use like tools like UCell or AUCell to evaluate the enrichment of that gene set of interest in different maps. For example, if you have another, in this case, the study, if you have another rat liver map, for example, we have like an immune enriched rat liver map and also a nuc-seq based rat liver map, and you could kind of like perform that enrichment in individual cells and then look if like the pattern is similar in this new map compared to what you kind of like identified in your original data. And then my last point is that preprocessing matters. Like be very careful and play around with different preprocessing approaches that you're taking. This is a great review paper, by the way, on the impact of data processing on integrative matrix factorization of single cell data. So check that out. And specifically when you're having multiple samples, like what I found that it's usually useful to normalize them individually, put them together, scale the whole thing, and then apply the matrix factorization method. That usually gives you better results, but it's something to keep in mind that it can make drastic changes in the factors that you learn. And my last point is that if you're interested in playing around with Barimix, I put a like very simple tutorial based on HTML. I'm going to put additional code here but I didn't have enough time for today. But in this GitHub repository, if you want to, it's a simple tutorial, you can just like go through it and figure out how to apply Varimax to your own data. So yeah, I think that's it for me. Thank you for listening. And I think I was 3 minutes ahead. Oh, that's it, sorry. So yeah, thank you. I think I'm done. And I'm happy to take questions. Let me. Okay, so Benjamin is asking, is it misleading to interpret too much about the clarity of the signal from presentation in 2 reduced dimensions? Would it make more sense to quantify the clarity of the signals with silhouette widths, neighborhood purity? Like, do you mean by clarity of the signal, do you mean matching between the covariate and the factor that has been learned? Could you elaborate on that a little bit?
No, I just specifically mean what you talked about with, you had a 2-dimensional representation saying the signal wasn't very clear. Is a specific slide. I'm sorry, I don't remember the number of the slide now, but it was a specific comment about 20 minutes ago. Sorry. Yeah,
Here?
it was right here with the Varimax rotation.
Right.
And my point was just, maybe I just missed it, but it just seemed like you were saying the before and after, the difference was, oh, look at the visual clustering. One makes this all very clear and the other one doesn't. And my point was, you're using a lot of dimensions to create this kind of 2-dimensional presentation and then saying, oh, look, one is visually clearer than the other. Is that the best way to do this, or should we use other criteria and parameters?
Right.
And what are those
Well,
parameters?
that's a great question. As far as I know, there aren't any like ready to use interpretability measures for factor analysis, like so that you would, use a specific metric to say, okay, my factors are more interpretable. But the whole point, as far as I know, for having a simpler structure and being more interpretable, like this is something that statisticians have kind of have agreed upon. For example, as I was saying here, like what does more interpretable mean? It basically, like, statisticians have like sat down and kind of defined some metrics, for example, for saying that if I'm getting a loading matrix similar to this that has a lot of zeros and has like a few features or genes with high scores, I'm going to consider that as more interpretable. But like, we are using a simple visualization here, but I would argue that if you go into like each of these factors individually, and like if you're worried about this being only one example, if you go into individual Varimax factors that was learned from based on this data individually and try to interpret them and try to like match them, like run the whole interpretability pipeline, it would work much better on this Varimax data. Like it's very obvious. Like if you see like some clear patterns like this, it's I would say that it's the green light for you that okay, this method is working. So it's a good proxy. It's not a very like robust quantitative metric, but I would say that in my experience it works well. But yeah, that's something that you could work on to come up with interpretability metrics that you can easily say that, oh, okay, this method is working better than, for example, NMF is working better than PCA on this data. But as far as I know, at least I'm not familiar with any of them. I can see that you sent a link here.
Yeah, that's just a link to Aaron Lund's package Bluster that does a lot of these diagnostics for clarity and
For clarity of factors?
RMSD, that kind of thing.
Is this for factors specifically?
This is
Because there are a lot of these metrics for clustering, for example. But for matrix factorization, I haven't seen like people specifically measuring interpretability and kind of coming up with a mathematical formulation for that.
Okay, cool, thanks.
Yeah, no worries. Any other questions? Okay, great. And can feel free to email me and like follow me on Twitter. I think if you follow Parnomic on Twitter,
Yeah, you
you'll
can
be able
find
to
her.
find me there.
You can find her on the Slack channel as well.
Yeah. Oh, yeah. I'm also on the Slack channel. Yeah, good point.
And
Okay, thank you.
all right, thank you, Dilaram.
No worries.
And thank you everyone for attending. I know it's summer. Everyone wants to go out. So we will end our session today. We wish everyone like a really good summer. And in the meantime, we will plan and we'll upload everything and we'll keep everyone informed. So that's it. Thank you. Right.